Morning, guys. We're going to be going over cell communication and signaling today. Cell communication is just how organisms are going to communicate within their body. You can also communicate out or externally, but we're going to more specifically look at how organisms internally communicate through different ways. We have a couple different parts that we're going to talk about. We're going to talk about the signaling molecule, the reception here of the signaling molecule to a target cell, and then that cell or that cell's cellular response there. There are three different types of cell communication. The first being cell-to-cell communication. This one is where the cells need to come in contact with each other. In animal cells, there's gap junctions, which allow molecules to freely pass throughout the membrane. In plant cells, that there is the plasmadesmata, which is like a tunnel between the plasma membrane and the cell wall. We also see this in the immune system where cells are going to come in contact with each other and they're going to create cellular responses. They could produce antibodies and then just a number of different things per each cell. This is juxtacrine is another name for the cell-to-cell communication. My analogy is it's like a paper note. You have to be in person. You have to be able in physical contact with that person to pass that information. The next is short distance communication. Again, it's a little bit further than the cell to cell. There's no contact here. Two different types. We have paracrine and synaptic. Synaptic is, if you're taking AP psychology, you probably know this word, the nerve cells and neurotransmitters. This is how our nervous system works. Even though the nervous system might take something from your foot to your brain, that is very long distance. The nerve cells are actually doing it within a short distance here. Paracrine is just when a cell secretes signaling molecules and other targeting cells are going to receive those and respond to that stimulus. Lastly, we have long distance, which is also called the endocrine system. This is where we're going to use steroids and hormones that are going to be created in organs, and they're going to travel throughout the circulatory system, and they're going to create cellular responses that way. The analogy that I give for this one is the Facebook post. This is where not everyone is going to react to the same way as maybe like the short distance one, or the juxtacrine, where the cell to cell communication is. I forgot to mention the analogy for short distance, which is like a Snapchat. Okay, this is going to be specific to a type of cell. It's going to respond in a specific way, and again, you're only targeting certain cells. With the long distance one, again, this is like the Facebook post. It's going to affect some cells, but not all cells. With signal transduction, and this is probably one of the more important parts of the chapter, we're going to talk about how cells are going to secrete some type of ligand, which is some chemical. Could be a protein, could be just a regular molecule compound, and it's going to go to a target cell. That target cell is going to receive it here with a receptor. You can see the membrane proteins that we talked about before. Whenever it receives that receptor, receives that ligand, it's going to change shape in some way, and it's going to create this cascade of different relay proteins that are going to create some cellular response. Now, the big thing here is we're going to target this to specific cells, so not all cells will have the same receptors. Think about your liver cells and your skin cells. They're going to have different receptors because they're looking to have different cellular responses, and you want that differentiation between your cells. There are 3 or 4 main steps within signal transduction. You're going to see it different in different places. I have 3 here. First is going to be your reception. This is where the ligand is going to bond with a receptor. Again, that receptor is a membrane protein. It's going to have a site that coincides with that signaling molecule. Now, whenever that reception occurs, it's going to change shape, which is going to maybe activate. It's going to send a molecule some way. This is just a very large overview. This is a very brief, hey, this is what happens in the cell. Every single type of signal transduction pathway is going to be different. So after this changes shape, it's going to create this signaling transduction where different molecules or different proteins are going to be activated, deactivated, changed, added to, in some way changed. And these secondary messengers are going to create some type of kind of cascade or some type of just relay of information. Our last part here is the response. This is where the cell is going to respond to that signaling molecule in some way. You can see here, make a protein, produce energy, enter mitosis. There are just a plethora. There are hundreds of thousands of these different pathways that we've found. There's just a ton of these things. The specificity of the signal is really important here. Again, we're just going to look at overarching ideas and overarching similarities. One of the things we do see, though, is one signaling molecule can either have one response or it can have many responses. I talk to my kids about epinephrine and how many different responses it has. Epinephrine is your fight or flight hormone, and we're not going to really go really in depth in this PowerPoint. But basically, it can raise your heart rate, it can raise how quickly you're breathing, it can slow down your digestion. It can make you sweat. There are many different cellular responses for epinephrine. So just understand that one signaling molecule can have a lot of different responses or just one different response. And there might be other signaling molecules that have very similar or the same responses in this way. We talk about the and or the or logic here with the signaling molecule. Sometimes a response will need two different ligands, or sometimes two different ligands will again create the same response. There are a lot of different types of signal transduction pathways, so they're all dynamic and they're all different in their own way. So a phosphorylation cascade is one type of signal transduction. You can see with this cascade, we're going to talk about kinases and phosphatases here a little bit. Kinases are molecules or specifically proteins usually that take a phosphate group from ATP and phosphorylate other proteins. That usually activates or deactivates them. Now, by phosphorylation, I mean just giving it a phosphate group. In this relay, you're going to have one kinase that activates another kinase that activates another kinase, and so on and so forth until we get this cellular response. Now, phosphatases do the same thing, but backwards. They actually remove phosphate groups. This again can activate or deactivate enzymes or proteins, and it's different between every single signal transduction pathway. But it's just one part that you have to know here. So I would know the kinases, phosphatases, and what phosphorylation is. Amplification of signal. This is what the cascades can do. So once one kinase is activated, it's going to activate other kinases or other molecules, and they're going to get this exponential growth of the cellular response. This is very good. It looks like this could be a problem, which it can be a problem, but this is really good for the cell. This means that the cell can respond very quickly to a stimulus. If you think about epinephrine, you want your body to quickly produce the cellular response because you are in a life or death situation. So this is just one way that the body has evolved different signaling pathways in order to survive. Now, there are three different type of receptors that we talk about a little bit in AP Bio. I remind my students, don't focus on these. I just want to tell you about them so when you go to college, you can say, oh, I remember talking about these a little bit. They'll go more in depth in college about these, and you'll have to know a little bit more. But I always like to talk about them just a little bit so you can say, hey, I definitely have seen these before. The first are G protein coupled receptors. These are when it's type of G protein that whenever it's activated, you can see here this signaling molecule is going to bond with a receptor. It's going to activate this G protein. The GDP molecule here is going to be replaced by GTP, which activates this whole protein, and it's going to send that molecule out to activate or deactivate other enzymes. But it's just called G protein. It's like a switch. Here's an image of it. You can see that GTP replacing the GDP and activating this molecule. Tyrosine kinase receptors are a type of protein where there's going to be two of them, and once the ligand bonds with both, these two receptors are going to come in contact with each other and form what's called a dimer. Once that dimer is formed, it's going to allow tyrosine, which is a amino acid, to bond with a bunch of phosphate groups. Once those tyrosines are bonded with a bunch of phosphate groups, they're going to create, again, cellular responses. Looks really complicated, and it is. All I just want you to remember is these guys are going to help with growth factors and helping cells enter mitosis. So you can see here, these are the tyrosine kinase receptors. You have your ligand up here. These ligands are going to bond here with these receptors. Once they bond, these two are going to come together and form that dimer and create a cellular response. Ligand-gated ion channels are our last type. This is where a receptor is going to bond with a ligand, and there's going to be an exchange of some type of ion through the membrane. We talk about a lot about this with neurons and the sodium-potassium ion pump, but these are just another type where you're going to get an exchange of material through the membrane. You can see here the ligand is going to bond. It's going to allow certain ions to go through the plasma membrane. Hormones are our last type of cell to cell communication, or it's the long distance communication. This is where certain organs are going to create these hormones that are going to go through the circulatory system. When they reach a cell, you're going to have sometimes they're going to dock with receptor proteins, but sometimes they're intracellular receptors, which means they're going to be a little bit smaller and nonpolar. They're going to cross that membrane protein. They're going to bond with an intracellular receptor, and they're going to create some cellular response. This is just another name for the endocrine system. So we're going to talk about diagrams a lot, and we're going to look at diagrams a lot in class. And one thing with diagrams is there's no universal way of how to interpret a diagram or how to make one. I've seen I've seen exceptions to these rules a lot. So to understand, this is just like a brief overview of what I've seen. Arrows mean that molecules are going to activate other molecules. So you can see here, this GLIF here is going to activate GLIR. I've also seen arrows show movement. So this could say, hey, this is moving to this location. So be wary of these diagrams. They'll usually come with like a synopsis or description. But what I've seen is these arrows most of the time show activation. Bar-headed lines are these lines right here. This shows that the molecule is inhibiting some type of process. So you can see here, pKa is inhibiting the GLIF from activating GLIA or becoming GLIA. So this is stopping that process. Our last concept that we have to understand for cell communication are feedback loops. Feedback loops help us either amplify or regulate certain cellular responses. In a positive feedback loop, the product here, B, is going to stimulate A. So you can see here, this is an analogy for that positive feedback loop. The number of cattle running is going to increase the panic level. The panic level is going to increase the number of cattle running, which then increases the overall panic. So you can see this is a loop that's going to go back and forth. And it's going to stimulate the other, which occurs until there's an outside stimulus that is applied to one of these loops. This helps maintain homeostasis, and this is good for the body. Again, we need this, but it also can be a problem if not regulated correctly. A negative feedback loop is when the product stops the initial stimulus here. You can see here B is going to stop A. I like the analogy of a thermostat here. Whenever it gets too warm in a room, or when it gets too cold, we'll say, the heat's going to kick on, which raises the temperature, which is going to stop the heat. So it's a feedback loop that's going to regulate how much of a cellular response there is.